Everyone, and welcome to our 151 exam review for exam 1. I'll be talking about units 1 through 3 in this review today. It should take approximately an hour and a half to 2 hours. We'll see how far we make it through. I apologize to those of you that tried to watch the last video. I did not have my sound turned on, so this is take 2. All right, so if you're here, it's because you're studying for exam 1 here at Chaminade University with the anatomy biology department, and we're going to talk today about the introduction to anatomy. So the objective for everything that's going to be on this exam in terms of this chapter is going to be a lot of definitions and a lot of terminology. So we want to talk about the different levels of structural organization. So this image is going to be important. So you'll need to know that the chemical level is the smallest level of organization, and it becomes more and more structured as we go up to the cellular level, the tissue level. Tissues come together to make organs. Organs come together to make organ systems, which obviously come together to make organisms. So you'll need to know the levels of organization. I think there is one question where all of these are listed and you have to tell me from the most complex to the least complex. So you basically need to know that at each of these levels, the whole is more than the sum of the parts. You need to know the 11 systems of the human body, including integumentary, so skin and hair and nails, skeletal, right, all of your bones, muscular, which is all of your muscles, nervous tissue, endocrine tissue, so the secretion of different hormones from glands is going to be the endocrine system, cardiovascular system, anything that's going to involve the blood and blood supply, lymphatic system, which is going to help take any of the extra fluid that's leaked out of the capillary bed back into circulation, but first run it through a couple immunity molecules like B cells and T cells to help eat all the bacteria and things that microbes that might have entered into it. Respiratory system, this is going to be a site of gas exchange. You'll note that the cardiovascular system and the respiratory system work hand in hand. Lots of these are going to work hand in hand. Cardiovascular and lymphatic work hand in hand. Skeletal muscle and muscular system work very well together. The nervous tissue is going to innervate the muscular tissue, which moves the bones. So all of these are going to work hand in hand. Digestive system is how we're going to be pulling in our nutrients. Urinary system is going to be how we are releasing our liquid wastes. A reproductive system is how we're going to create new offspring and new individuals. All right. So we talked about the different life processes that separate life from non-life. Life processes include metabolism, responsiveness, movement, growth, differentiation, and reproduction. And even the very smallest of organisms display all of these. So even if something as simple as bacteria growing towards a sugar in a growth media, or a plant turning itself towards light, those are considered a responsiveness and motion, right? Every organism is going to grow and eventually end up dividing. With multicellular organisms, we also have differentiation. So cells that go from an unspecialized state to a more specialized state and perform a specific function. You've heard me say homeostasis a million times, and you'll hear me say it a million more. But basically, with homeostasis, we want to make sure that we're maintaining the internal conditions of equilibrium within the body. And for every single parameter that you can think of, every single nutrient, there's a set of parameters that are going to be too high and too low. Again, I told you this is going to be this blank slide, essentially, where this is going to be an overview, but when we see this later on throughout the semester, we're going to be discussing specific controlled conditions that are disrupted in specific ways. But this negative feedback loop is something that you're going to see quite often. Basically, what happens is you have some sort of external stimulus that disrupts homeostasis by changing, increasing, or decreasing whatever this controlled condition is. All the controlled conditions in your body are monitored by specialty receptors. Chemical receptors monitor your chemical balance. Tactile receptors measure touch, right? Stretch receptors measure stretch. Baroreceptors measure blood pressure. Lots of different receptors in your body measure a lot of different controlled conditions. And when those change or go haywire, they're going to send information. The receptors send information to the control center. This is almost always the brain. And then the brain can send information out to the effector cells that are going to cause a change that will then cause a response. Once that response alters that controlled condition, hopefully it's going to restore this back to homeostatic conditions. Okay. We also talked about fluid balance, how we have 3 major types of fluid. We have fluid that is inside the body cells. That fluid is called intracellular fluid. You may also hear me call it cytoplasmic fluid. We have extracellular fluid or interstitial fluid. Extracellular fluid is found in 2 major places, right? It's going to be found filling all of the cells. That's called interstitial fluid. We also have blood tissue plasma, which is called called blood plasma, but it's another type of extracellular fluid. And inside lymphatic vessels, this fluid is called lymph, which is going to then feed back into the vasculature system after being fed through things like lymph nodes to help destroy any foreign microbes and bacteria. All right, so again, homeostasis is going to be, oh, one last type of extracellular fluid is going to be fluid that's going to be surrounding the brain and spinal cord. That's going to be called cerebrospinal fluid. We also have fluid in the synovial joints called synovial fluid, and aqueous humor or vitreous humor in the eyes. And all of this exchange between the fluid that the blood and the lymphatic tissue, from the interstitial fluid to the cytoplasmic fluid, and then back from the cytoplasmic fluid to the interstitial fluid to the blood plasma, is basically this gradient of difference of the concentration of solutes in these three fluids is how we are going to exchange things into and out of the cell. Okay, so how do we maintain homeostasis? Anytime we get out of balance, we want to get back into balance, and we do that by these general feedback systems. Most of the time we're dealing with negative feedback systems. So let's talk about negative feedback systems. So negative feedback systems, right? We have a stimulus disrupting a controlled condition. In this case, the controlled condition, so we're given real information here. The controlled condition that we're disrupting is blood pressure. Now remember, every single one of the controlled conditions is detected by receptors. These receptors here are called baroreceptors. The baroreceptors are going to detect blood pressure and blood vessels. That information is then going to send to the control center. Again, this is the brain, which is then going to send information to the effectors, the heart and the blood vessels. This causes a response. Hopefully this returns us back to normal. In this case, the response is going to be dilation or widening of the blood vessels. And also the heart is going to have a decrease in heart rate. These two together are going to combine to cause a decrease in blood pressure, which is going to return us back to homeostasis. So that's going to be a negative feedback system. Anytime we get too high, we want to bring it back towards center. Anytime we get too low, we want to bring it back towards center, right? Sometimes we have positive feedback systems, and you're not going to see these very often at all. In fact, you probably only see this particular one. But basically what happens with a positive feedback system is we have an aberrant condition that we want to alleviate. For example, we have a baby that we want to deliver, or we have a splinter in our toe that we want to force to the surface to rupture. So in this case, disrupting the controlled condition further disrupts the controlled condition. What do I mean by that? So in this case here we have stretching of the cervix, which is occurring because the baby is pressing on the nerve cells in the cervix. That information is sent from these receptors to the control center of the brain. But in a negative feedback loop, the brain would say, okay, well, get off of those or stop stretching, right? But in a positive feedback loop, it's going to encounter more stretching because it's going to cause the muscles of the cell walls of the uterus to contract, which causes more push on the cervix, which causes more stretching. So the body stretches the cervix more, increased stretching causes more oxytocin release, which causes more contractions, which causes more stretching, which causes more contractions, more stretching, contractions, and stretching, until finally something changes, till something actually disrupts the cycle, like the birth of the baby or the splinter rupturing through the surface of the skin. So a positive feedback system is going to strengthen or reinforce a change in the body's controlled conditions. All right. If we end up out of homeostasis, we can end up with disease states or sometimes even death. And we label these in terms of how severe our issues are. So a disorder is when we have an abnormality of a structure or an abnormal function. But disease is when we have a series of symptoms, recognizable set of signs and symptoms that are associated with that disorder. We can have different types of diseases, like localized diseases affecting one region, systemic disease that affects the entire body. And we have differences between symptoms and signs as well. So when you're going to run an interview with the patients, you need to make sure that you're recognizing the difference between things that we can test, like temperature, right? Things that we can observe and measure, signs, fever, rash, etc., temperature increase, versus symptoms, which are things that are not necessarily apparent to an outside observer. I have a headache. My stomach makes me feel a little bit nauseous or queasy, right? These are things that you want to take into consideration, but aren't necessarily signs, because these are relying on the patient to provide accurate information. All right, so once you take all of the information that you can and you've assessed the situation, then we end up taking all that information, synthesizing it, and making what's called a diagnosis. And this is how we're going to distinguish one disease from another, by looking at the nature of the disease. And we're going to do this with a lot of information. So you're going to take the medical history of the patient, administer the physical examination, and then usually we're going to corroborate this diagnosis with some sort of medical imaging technology. All right, so what is on the exam in terms of body positions? So in anatomical position, you're going to have your hands down and your palms facing forward. Your feet are going to be flat on the floor with your toes facing forward. If you are laying down in a reclining position, when you are face up, it is called the prone position. I'm sorry, if you're face down, it is the prone position. I apologize. If you are face up, it is called the supine position. There is a question on that, so do make sure that you know the difference here. Regional names are going to be given for specific regions of the body. So cephalic refers to the head, cervical refers to the neck, thoracic refers to the chest or the trunk area. We have the upper limbs and the lower limbs that refer to the arms and the legs. We also have a lot of different locational names, so do make sure that you know these regional names referring to different spots within the arm. For example, front of the elbow is antecubital, back of it is olecranal because it matches up with the olecrana of the ulna, which runs here along the back of the arm. All right. Again, I'm not going to go through all of these here, but you do need to know them. Any of them are fair game. So make sure that you study them. Everything that's on this list, totally fair game for the exam. All right, in terms of directional terms, you'll need to know where things are relative to one another in the body. So if something, for example, on the upper limb, if something is further away from the body, it's considered distal, closer to its proximal. So the shoulder is proximal to the elbow, right? And the elbow is distal to the shoulder, or the wrist is distal to the elbow. And if we're going towards the center, right, towards the sternum and the midline of the body, that's going to be called medial. If we're going away from it, it's lateral. So the shoulder is lateral to the nose, which is medial, right? If we're going upward to the body, it's like the head is going to be superior to the neck, right? The pelvis is inferior to the heart. So inferior means lower, superior means upward. Again, you're going to need to know all of these directional terms. So anything here, totally fair game. Same thing with planes and sections. You'll need to know how the different planes are going to divide the body. The way I like to remember this is what happens if you were dissected by these planes. So a transverse plane is going to cut me off at the knees. It's going to cut my legs off. Now it could also cut off at the trunk, right? Transverse just means it goes this direction. So it could cut up here at the shoulders, etc. But I like to remember it as it's going to cut my body in half like a magician, right? A frontal plane is going to cut your nose off. It's going to cut right in between your ears and take the front of your face off from the back of your head. Right? A mid-sagittal plane is going to go directly through the midline of the body. A sagittal plane is just going to be in this direction, but a parasagittal plane is going to be 1/4 of the way. So there's a parasagittal line that's going to go through the knees and through the toes. But if you were to happen here, for example, this would just be considered a sagittal slice. And an oblique plane is going to go through at any random direction. And usually when you're given an oblique plane, it will also come with information, oblique plane of 45 degrees or whatnot, to tell you exactly what direction it's being sliced through. So again, anything here, totally fair game. All right. You also need to know when we're looking at images what your view is. So what kind of section we take is going to determine what your view of the organ is. Here's 3 different views of the brain, the mid-sagittal, frontal, and transverse section. And again, if you didn't know, they would look like they were completely different organs. So you have to make sure that every time that you're looking at an image, you're orienting yourself both with the plane and with the view that you're taking. Okay. Know all of the major body cavities. So you'll need to know cranial, vertebral cavity are going to have the cerebrospinal fluid. Thoracic cavity is going to include the pleural cavity and the pericardial cavity, which are going to be the lungs and the heart. The mediastinum is going to extend from the sternum all the way back to the vertebral column. And then we also are going to have the diaphragm right here, which is going to be separate out the thoracic cavity from the abdominal pelvic cavity. Abdominal pelvic cavity is going to be one large region, but the pelvic region is going to be separated out by where the pelvis lies. So anything below the pelvis is considered the pelvic cavity. Then in the pelvic cavity, we're going to have our reproductive organs, our urinary bladder, etc. In the abdomen, we'll have our stomach, our spleen, liver. So a lot of these organs are going to be separated from each other by these major cavities. So again, make sure you know which is which. Pleural cavity is going to surround the hearts. Pleural cavity is going to surround the lungs. The pericardium is going to surround the heart. So the pericardial cavity is going to surround the heart and the pleural around the lungs. I apologize for that misspeak. I have said this a lot in the past couple of weeks. All right. So again, a transverse plane. In this case, we're looking at an inferior view, so we're looking upward of a transverse plane. Again, this would cut your legs off, except in this case, we're going straight through the chest. And we're looking into the thoracic cavity. If we're looking at our cavities, we have 2 pleural cavities, right, that are going to surround the lungs. And then the mediastinum, which is going to also include that pericardial cavity, which surrounds the hearts. All right. So the abdominal cavity is going to be divided into 2 regions, superior and inferior. The superior is the abdominal, the inferior is the pelvic. Again, the pelvic is where we're going to have our urinary bladder and our reproductive structures. And in the abdominal cavity, we'll have our stomach, spleen, pancreas, and accessory organs of digestion like the liver and the gallbladder, etc. Let's see, moving down. We are also going to have membranes that separate these out. We have serous membranes that separate out the major cavities, and you'll need to know the names of all of these different cavities and their membranes as well. So you'll need to know, for example, the names of the membranes that separate. So diaphragm is going to be the membrane that separates out the abdominal cavity from the thoracic cavity, for example. All right. Also, so the abdominal pelvic regions and quadrants. So we're going to have 9 major regions that are drawn basically by drawing a tic-tac-toe board. And when we look at each of the organs, they are each found in one of those 9 quadrants. And so you kind of want to know where the major organs are, because one of the first things that happens when a patient comes in with a problem is they're going to say it hurts here. And you're going to need to know what organs are in that location so that you can accurately make a diagnosis once you get the rest of the medical history. The medical imaging, I have not added any of the images for. You will probably need to know them if that's something that you go into, if it becomes your field, but it's going to be a specialty kind of thing. So if you go into radiology, you're going to have many classes involving how to use and go through how all of this works. So I do not have a ton of questions on these. However, there are 1 or 2 questions that might ask you, and a patient comes in with this problem, a broken bone, for example. Which of these do you think would be most likely to detect whether the bone is broken? So you might, or a woman comes in with a pregnancy, which one of these would be the most likely one that you'd use? So you would pick ultrasound, right? So it basically depends on what you're looking for and what kind of diagnostic technique you're going to use to be able to determine what you need to see. So that's going to wrap up Unit 1. I'm going to move on now to Unit 2, or Organization of Matter. This is going to be referring to basic chemistry. So chemistry is going to be the science and structure of any of the interactions of matter. Anything that occupies space and has mass is considered matter. And remember, mass is going to be the amount of matter that's involved, whereas weight is going to be gravity acting on that mass. So it's going to be a little bit more complicated because your weight is going to be different in a different place that has a different gravity, like on the moon, whereas the mass would remain the same. All right, so we have 3 major forms: solid, liquid, and gas. Picture this as ice cubes and water vapor. All matter is comprised of chemical elements. The ones that we're going to be talking about mostly are going to be oxygen, carbon, hydrogen, and nitrogen. These are going to be the major body's elements. We have 96% of the body weight. Again, you're not going to need to memorize how much of what are going to be where, but you need to know that these are the 4 major ones, and you need to know how they interact. So you need to know carbon can make 4 bonds, hydrogen can make 1 bond, nitrogen can make 3, oxygen can make 2. You'll need to know that, and as going forward, I'll explain exactly why that is. Okay, when we break down these molecules into their atoms, again, the atoms are going to be the smallest unit of that matter that retains the characteristics of the element. Anything smaller than that are going to be subatomic particles, and they're going to be the same no matter what atoms you look at. So an atom is going to be comprised of 3 major particles: the protons, the electrons, and the neutrons. The nucleus is comprised of the protons and the neutrons. Protons are positively charged, neutrons are neutral, electrons are negatively charged. But when we carry weight, protons and neutrons each have a weight of 1, and electrons are going to have a weight of 0. We have an electron cloud model where we're showing all the electrons bouncing around the outside, but to simplify that, we should show this as an electron shell model. Electrons are organized into these shells. The innermost shell is going to have 2. As we proceed outward, they're going to expect to have 8 electrons to be considered full. This outer shell is called the valence shell. In this case, we know we're dealing with carbon. Why? Because we have 4 outer valence electrons. And when we have 4, that means we can make 4 bonds, okay? Because again, we want to get up to 8. So we can make 4 single bonds, or we can make 2 double bonds, or any combination thereof. Again, our subatomic particles are going to be comprised of protons, positively charged with a weight of 1, neutrons uncharged with a weight of 1, electrons negatively charged with a weight of 0. We have a nucleus inside the atom, again, that has the protons and the neutrons in it. The electrons are going to be outside of it in the shells or in those well, it's going to be an electron cloud, really, but to simplify it for our understanding, we would depict it as an electron shell. So we also need to know the atomic number and the mass number. So the atomic number is going to be the number of protons, and the mass number is going to be the amount of protons and neutrons together. So for carbon, the atomic number is 6, but the mass number would be 12 because the protons and the neutrons, each of them weigh 1, and there's 6 of each, so that's 12. So our atomic mass is going to be 12. All right. So again, this outer shell, this valence shell, is going to have 4, which means it's able to make 4 bonds. That can be any combination of 4 single bonds to 2 double bonds, etc. Okay, we do have specialty types of isotopes. So isotopes are going to be atoms of an element. They're going to have a different amount of neutrons, but the same amount of protons. Some of them are stable and are not going to change over time. Some of them are called radioactive and are unstable, and they're going to start to decay. And this is how we are able to carbon date things, for example, because we know the relative concentration of these different isotopes, and we know that it changes over time as we enter into decay. And so we can tell how old something is by measuring the percentage that of the carbon that are in these particular isotopic forms. All right, again, atomic mass is going to refer to the weights of these subatomic particles. Neutrons are going to have a mass of 1, protons a mass of 1, electrons a mass of 0. Anything after the 0.00 we're going to just discard. Okay? for the purposes of this class anyway. In chemistry, they'll need to calculate that out, I'm sure. Now, we have ions, molecules, and compounds. So anytime an atom gives up or gains an electron, it becomes an ion. So an atom walks into a bar, he says, I've lost an electron. The bartender says, are you sure? He says, yep, I'm positive. Get it? Because he's become a positive ion, he's lost an electron. Anyway, you're going to end up with a positive or a negative charge because you have an unequal number of your protons and electrons. And you might do that because you want to settle your valence shell. So if, for example, you have 7 in your outer shell and another one comes by and has 1, it might be easier for the guy that has 1 to just donate that 1 electron to the guy that has 7, making it have 8, and the other one have 0, and that will make it happy, make both molecules happy. So that's how ions are created, like sodium and chloride, for example. Now that brings us to chemical bonds. When we have atoms such as sodium and chlorine, which have the ability to donate or accept electrons, again, they become ions. So here's sodium. Sodium is going to have 1 electron in its outer shell, so it's going to donate that to chlorine. Chlorine has 7 valence electrons. It picks up 1, it has 8. This one's going to go down a shell and have 8. They're both going to be happy. That's called an ionic bond. This is when we are transferring electrons, maybe not permanently, but for a stable way for an indefinite period of time. That's called an ionic bond. And when we do this, one ion is going to be a cation and one is going to be a negative, cations are positively charged, anions are going to be negatively charged. So this is going to be an anion, that one's going to become a cation. Okay? Now, sometimes these ionic compounds are going to dissolve. Most of the time they're going to dissolve into solutions like water, for example, which is going to create an electrolyte solution. So a solution of a liquid that's going to have positive and negatively charged ions is going to be called an electrolyte solution. Now, as I mentioned previously in class, water is kind of a promiscuous molecule, and water with its partial positive and partial negative charges can disrupt these dance partners. And that means that we're going to dissolve sodium and chloride into water. That means that ionic bonds are not going to be useful in biological systems because we have a lot of water in biological systems, right? We are entirely aqueous solution. So the kind of bonds that are going to hold well in water or an aqueous solution like our blood supply are going to be covalent bonds. So covalent bonds are going to be formed by sharing electrons. So instead of transferring them, they're going to be shared equally or unequally. If they're shared equally, it's called nonpolar, such as these 2 hydrogens each sharing 1 electron with each other. Now they're happy. These 2 oxygens each sharing 2, they're all happy. Nitrogens each sharing 3, all happy, right? However, water down here at the bottom, we're going to have 1 hydrogen bound to this oxygen and 1 hydrogen bound to the same oxygen. So 2 hydrogens bound to the same oxygen, but they're going to be sharing different sets of electrons is my point. So when this hydrogen binds here, it's sharing that electron. When this hydrogen binds there, it's sharing that electron. But remember, just like the lawnmower example that I gave in class, which I don't really have a lot of time to get into now, but if these guys are sharing a lawnmower and this is a smaller lawn, the electron's going to spend less time, the lawnmower is going to spend less time mowing this lawn and more time mowing that lawn. Now, if your lawnmower is negative and it spends more time here, it's going to give you a partial negative charge. And the electron being negative, it's spending less time here, gives it a partial positive charge. So hydrogens are going to be partially positive, oxygen's partially negative. And that's going to account for what we call hydrogen bonding properties of water as well. And it's also going to allow it to become a little bit of a promiscuous molecule in that all the negative charges can align around a positive molecule, or all the positive charges are aligned around a negative molecule, and pull sodium and chlorine away from each other in aqueous solution. Okay, so again, covalent bonds are either nonpolar or polar. If they're nonpolar, they're shared evenly. If they are polar, they're shared unevenly. And when they're unevenly shared, we end up with partial positive and partial negative charges. That allows us to create hydrogen bonding. Now, hydrogen bonding occurs between atom A and between molecule A and molecule B. I want to be really clear. It does not occur between the atoms in the same water molecule. It occurs between the atom hydrogen here in this water molecule and the oxygen atom in that water molecule, or the hydrogen and this oxygen. So it's going to be from 2 neighboring molecules, and this allows for the cohesive nature of water, or for water to attract to one another. This allows for all of the water at the roots of the tree to make it all the way up to the top of the tree, because every time the water is going to be pulled up from the top, it's going to pull the molecule next to it, and in that way the whole thing is going to be sucked up kind of like a straw. Okay, so hydrogen bonding in and of itself, these are weak bonds, but as a whole it can become a very strong force. Okay, it gives us cohesion, creates surface tension of water, etc. All right, so now let's talk about chemical reactions. Anytime we make new bonds or break old bonds, that's called a chemical reaction. And anything that we start with is a reactant or a substrate, and anything that we end up with is a product. Okay, reactants, products. But I do want you to notice that although we are changing from 2 hydrogen gases and 1 oxygen gas to 2 water molecules, we are keeping the same amount of atoms. Okay, still 4, 1, 2, 3, 4, and 2, 1, 2. So we are converting things, but the total mass of the reactants is still going to equal the total mass of the products. So that's going to be the conservation of mass. Okay, metabolism is all of the chemical reactions, including the catabolic and the anabolic reactions that occur within an organism. So all the ones that build you up as well as break things down. All right, so we have energy as well. Energy is denoted as the capacity to work. We have potential energy, which is energy that is stored. Picture a diver on a springboard about to jump. Um, or energy in a battery, for example. Kinetic energy is energy of motion, so the energy of the roller coaster actually going down or the diver actually jumping off of the board. And chemical energy is going to be potential energy, but it's potential energy stored in compounds or molecules. So stored within the bonds of, for example, ATP, which is one that we use a lot. So we have two major types of reactions: reactions that release energy, or exergonic reactions that energy exits the system, or reactions where energy needs to enter the system, an endergonic reaction. Okay, and we use ATP as a carrier molecule between these two reactions. So in the cell, we constantly have exergonic and endergonic reactions. Exergonic reactions release energy, and ADP and free phosphate are sitting right there ready to be formed into ATP. And then ATP roams around the cell until it finds a region where an endergonic reaction needs energy. It breaks that third phosphate off to diphosphate and releases the energy in order to be able to drive this endergonic reaction, such as binding amino acids together to form proteins. Okay, catalysts are enzymes or biological catalysts or chemical compounds that speed up the rate of a chemical reaction. They do that by lowering the activation energy, and keep in mind that the catalyst itself remains unchanged. So even though it can change throughout the reaction, at the end of the reaction, it's going to remain in the exact form that we saw at the beginning, so that it can then lather, rinse, repeat another set of substrates to turn them into products. So it's going to work over and over and over until it is told not to do so anymore. So we have two major types of chemical reactions: reactions that build things up and reactions that break things down. Synthesis and decomposition reactions. Other types of reactions include exchange reactions, where we're exchanging one for another, one atom or a series of atoms for another atom. And reversible reactions mean that we can take our product and turn it back into our original molecules. And most reactions are reversible. However, oftentimes we have a cascade where we are going to create A into B and B into C and C into D. And in that way, we eventually end up with Z, which is not able to be converted immediately back into A, right? Redox reactions or oxidation reduction reactions are going to happen anytime electrons are transferred between atoms and molecules. So remember that Leo goes Gur. So loss of electrons is oxidation. Loss electrons, oxidation. Gain of electrons, reaction. So Leo the lion goes Gur. But these are going to happen hand in hand most of the time. That's why we call them redox reactions, because every time that we have an oxidation, we also have a reduction, right? There's no way to lose an electron without someone else taking it from you. So redox reactions happen constantly in your body. And they happen two different reactions at the same time. One loses the electrons and one gains them. Okay, so let's go back to the water, the diagram of water for a second. Water is really important because it's polar. What does that mean? That means it has an unequal sharing of electrons. The lawnmower spends more time on oxygen than it does on hydrogen's yard. That means that hydrogen is going to have partial positive charges, and oxygen with that negative electron a little bit longer has partial negative charges. Okay, this is our structural formula. It's also known as H2O. And water is really important because it is the universal solvent. So as I mentioned previously, it's able to take all of its partial positive charges and line them up to create partial positive charges and line them up to create a positive charge. We can dissociate chlorine, right? The equivalent of one positive charge will pull chlorine and its negative charge away from sodium. And we're going to do the same thing with other water molecules here with all the oxygen atoms and their partial negative charges lined up around sodium. And this way, water can be kind of promiscuous and pull these dance partners away from each other on the dance floor. That means that salt is going to dissolve pretty much immediately into a water solution. Okay, so a solution is comprised of a solvent and a solute. Solvent being the liquid, solute is what goes into it. We can also have other types of things like a suspension. Picture putting sand in water or glitter in water. It will settle out eventually and is not going to ever completely be suspended in solution. Whereas in the solution here, right, the solvent is actually going to dissolve the solute. So we're going to actually end up with the crystals of sodium chloride going into hydration, right? So hydrated sodium ions and hydrated chloride ions. All right, so why is water so important? So water is important because it is used to create a reaction. So it participates in all of the biological reactions in the cell. Anytime we are creating, so it's going to take two smaller molecules and make one larger molecule. Here we have two monomers making a dimer. We're going to do that by dehydration synthesis. And in order to do that, we have to remember the example I used in classes. In order to make two students hold hands, I have to take their coffee cups away first, right? So first we're going to remove this hydrogen from here, that OH from there. So now no one's carrying coffee, so they're able to hold hands. That's going to create a water molecule and create a dimer. So that's called dehydration synthesis. And the opposite of that, if we were to take this sucrose molecule or a dimer and break it down into its monomers of glucose and fructose, we would go ahead and separate out here at that oxygen, and then we would lyse this water molecule called hydrolysis so that we have one OH group and one H group to cap off the sticky ends. We're essentially putting the coffee back in the student's hands. Okay, so hydrolysis is when we're breaking large molecules down into smaller ones, and dehydration synthesis is when simple molecules are coming together to create a larger molecule. Okay, again, I just spoke of the difference between a solution and a suspension. So a mixture is anytime we have something that's physically blended together but not bound by chemical bonds. A solution is when something is dissolved. Usually there's more solvent than solute in a solution. So usually there's more water than salt, for example, right? A colloid is going to be when we have things that are suspended, but because of the size of the particles, things are going to end up, make sure that's like an emulsion, something like mayonnaise. In suspension, the material will mix with the liquid, but will eventually settle out after a little bit of time, something like glitter in water or sand in water. Okay, and just like there are two different ways or many different ways to talk about the weight of something, right? We can talk about it in pounds or kilograms. We can also talk about the concentration of solution in two different ways, percentage and molarity. So percentage is going to be the relative mass of the solute found in the given volume of solution, 1 gram in 100 milliliters. But a mole is going to be relying on using the molecular weight or the atomic weight of that element to put exactly 1 mole of that element in there. So for sodium chloride, that's 58.4 grams. So if we put 58.4 grams in a liter, we have a 1 molar solution. So a molar solution would be talking about the weight of 1 mole of that chemical in 1 liter of solution. And again, that's going to rely on the molecular weight of that particular chemical. Okay, we also have acids and bases and salts. So let's go down here a little bit and talk about that. So a salt is any time that we have a positive and a negative ion that come together, like KCl, but we are not including a hydrogen or an OH group in there. So when it dissociates in water, we're going to have an even amount of positive and negative charges and no change in the pH because we don't have any change in the concentration of OH or H. Now, something like a base, where we're going to have potassium, which is the positive, the OH is negative, so we're still going to have a positive and negative charge added to solution. So we're not going to change the charge of solution, but we're going to change the relative concentration of OH relative to hydrogen because we're adding more of the hydroxyl group. An acid will add more of the hydrogen ions, right? Like something like hydrochloric acid. All right, so acidity and alkalinity is based on a pH scale, which runs from 0 to 14. It is a logarithmic scale, so it's not linear. What does that mean? That means that we go from -7 to -6, or from -7 to -8, we have either decreased or increased a factor of 10. So it's actually tenfold difference. So if this is 10 molecules, this is 100 molecules. So actually 10 molecules, 100 molecules, 1,000 molecules, right? 10, 1, 0.1, 0.01, right? It's a tenfold difference, each of these steps. And again, we're looking at the relative concentration of hydrogen ions to hydroxyl ions in solution, right? And most of the solutions in our body are going to be somewhere between 6 and 8, with the exception of our stomach, and ladies, our reproductive tract, somewhere around 4. Okay, so the pH system is really important. We have to make sure that we maintain our pH, and we do that by specialty things called buffers. So just like anything, we need homeostatic conditions of pH. Too high is bad, too low is bad, right? And we use buffers such as bicarbonate to act as weak bases and weak acids to help accept either the hydrogen ions or the hydroxyl groups, depending, so that we can pull them out of solution to help keep our pH closer to normal. All right, so let's talk about carbon and some organic compounds. So carbon is going to be really special because it can react with a lot of other carbons. It can make large molecules that have many different shapes. Typically we're going to see rings, we'll see chains, lots of different carbon units that are held together by covalent bonds from one carbon to another. So this carbon backbone or carbon skeleton can then have things hanging off of them, which are called functional groups. And these functional groups which dangle off are going to be reactive groups. So typically the carbon skeleton is going to be like the backbone, and it's going to be less reactive than the things that hang off of there. And the different types of functional groups, you're not going to need to memorize all of them, but are going to include things like alcohols or ketones or aldehydes, right? And or sugars, etc. So what hangs off of these molecules are typically going to be the reactive groups or the functional groups. All of the organic molecules are going to be combined by monomers coming together to make dimers, dimers coming together to make polymers. So a single unit of a train car would be a monomer. Two monomers come together is going to be a dimer. Again, that happens through dehydration synthesis. And when we take that dimer and break it down into two monomers, we do that by hydrolysis. Okay. And if we have the same molecular formula but a different structure, picture left hand, right hand, right? They couldn't overlay exactly, but they have all the same digits. Those are called isomers. So sometimes we can have an isomeric form of the same molecule, which we may or may not be able to digest as well, right? Um, all right, so let's talk about the different types of biomolecules. We're going to talk about carbohydrates. Again, all of these different biomolecules have the monomer, the dimer, and the polymer. For carbohydrates, the monomer is going to be a monosaccharide, the disaccharide is the two, or the dimer, polysaccharide is three. Monomers can be either pentoses, or 5 carbon rings, or hexoses, 6 carbon rings. And they're going to get put together again by the process of dehydration synthesis. So dimers include things like lactose, which is galactose and glucose, or maltose, which is glucose and glucose, or also sucrose is another dimer. So a dimer is anytime that we have, so that's glucose and fructose is going to make sucrose. Anytime that we have two monomers coming together to make a dimer, again, that happens through dehydration synthesis. And when we split back into simple sugars, we do that by hydrolysis. Now, when we go from dimers into polysaccharides, now we're talking about much longer chains. So here's an example of a polysaccharide. Each one of these little hexamers is a glucose monomer. Principal polysaccharide is going to be a molecule called glycogen, which again is comprised of a ton of glucose monomers. And we can steal monomers of glucose from glycogen when our blood sugar gets too low to restore our blood sugar levels. If, for example, we haven't eaten but we started exercising, or if we've just eaten and we're not exercising and our blood sugar gets too high, we can take it out of solution and store it here in glycogen in this long polysaccharide version. Okay, lipids also are going to have a monomer and a polymer, a monomer, dimer, and a polymer. So in this case for lipids, the monomer here is going to be our fatty acids. So here we have a saturated fatty acid. Remember, all of the seats on the plane are taken. Unsaturated means that we have one kink in the tail because we have one double bonded carbon. And typically these fatty acids can then be connected to glycerol. If we do that, we can make something called triglyceride. So here's a triglyceride. These are going to be our energy storage molecules. Again, we can either be fully saturated, right? No seats left on the plane, or we can be partially unsaturated. It's considered a monounsaturated because we have one double bond that could potentially be broken open to have two more seats for hydrogens on this plane, right? Triglycerides or fat molecules are going to be your energy storage molecule. They're also involved in insulation and protection. They can be either solids if they're fats or liquids if they're oils. Liquids are going to be the liquid triglycerides are going to be better for you. Things like olive oil, for example, over baking grease. And the reason that is because it has to do with the percentage of unsaturation. So more saturated, these guys are going to stack more tightly on each other and they can cause heart disease. Heart healthy fatty acids are going to be unsaturated, at least mono, possibly diunsaturated. Something like I said, like an olive oil or a vegetable oil. Triglycerides are going to provide a lot of energy per gram, 9 as compared to 4.5 for carbohydrates and proteins. So it's more than twice the amount of energy that we get from carbohydrates and proteins. And so fat's going to be a really high energy source for us to be able to utilize under conditions of low food. However, under conditions of high food, if we have excess of carbohydrates, proteins, etc., they're going to end up deposited in adipose tissue or fat tissue as triglycerides. So it's going to be, again, your energy storage molecules. Triglycerides have glycerol in the head and the fatty acid tails, and they're held together by covalent bonds. All right, phospholipids we saw very frequently. Phospholipids have a polar head, non-polar tail. It's called an amphipathic molecule. Again, these can be either completely saturated or unsaturated, depending on the type of tail. But typically what's going to happen is we're going to make a bilayer. So we have an upper layer and an inner layer. We're going to have a series of polar heads interacting with aqueous solution, and then the nonpolar tails interacting with each other, kind of dangling towards the center. This is going to form a barrier. So we have a nice barrier function in this phospholipid membrane. Also embedded in that membrane, we're going to have some steroids, including testosterone, estrogens, cholesterol. Tons of cholesterol are going to be embedded in the membrane. Other examples are going to include cortisol. So a lot of the steroids in the body, some of them are sex hormones, but a lot of them are not, including cholesterol. They're known as sterols. Why? Because we have this ABCD structure. We have 3 hexamers and a pentamer. And then you'll see we have different reactive groups that dangle off, but this major 4-ring structure is what identifies it as a sterol Well, in this case, it's a cholesterol, right? You can see sterol right on in there. Other lipid molecules are going to include prostaglandins, leukotrienes, etc., involved in inflammatory responses and help in regulation of the body temperature. Leukotrienes are going to help again with allergic and inflammatory responses as well. And some body lipids are going to include some of your fat-soluble vitamins. Beta carotene is a good example. Vitamins D, E, and K are good examples, and lipoproteins. All right, so let's move on to proteins. So proteins are going to help do all sorts of things. Remember, they're the blue-collar workers of the cells, and their monomeric form is going to be an amino acid. An amino acid is going to be comprised of an amino group on one side and a carboxylic acid group on the other side with one hydrogen. This R stands for reactive, and that reactive group is a side chain. It can do all sorts of things. It can be really small, like glycine, just hydrogen. It can be really long, like lysine. It can have a bulky molecule in the middle, like this hexamer ring. Or proline, for example, has a pentamer ring, which causes a kink. So no matter what, wherever proline is, we're going to get a kink in the protein. Cysteine has specialty regions, kind of like Velcro. This sulfur can find another cysteine elsewhere and form a disulfide bond that will allow adhesion between one cysteine molecule and another cysteine molecule that might not be anywhere near each other in the actual sequence, as long as they're localized near each other in the final folded protein conformation. All right, so again, just like anything else, we're going to put these together by dehydration synthesis and break them apart by hydrolysis. So we're going to remove this OH group and that H group from, so the H group comes off the amino group, the OH from the carboxylic acid group. That's going to result in water and a peptide bond. And we can put polypeptides together. Again, we're going to start with a monomer of an amino acid, then we end up with a dipeptide, we end up with a polypeptide, and eventually we end up with a protein. These polypeptide chains can have up to 200 amino acids on them. They can be extremely long, and they have multiple levels of structural organization. So the levels of structural organization are going to start with primary, just the sequence of amino acids in order, to secondary structures such as your beta pleated sheets. Here's like a corrugated cardboard, or your alpha helix. Both of these you can consider, for example, this is a spiral staircase. You can consider that not only are you holding hands with the people that are directly in front of you and behind you in line, but you're also holding hands with people who are directly next to you in space. So for example, you're reaching up a staircase and down a staircase to hold hands with the people above and below you in space. And that's going to keep this conformation of the spiral. Same thing with the beta pleated sheet. You're not next to this individual amino acid in line, right? You might be jumping ahead 5 or 6, but you're next to them in space, and that allows you to hold that hand as well. So that gives you these secondary structures, all of the combination of secondary structures. So all of the beta pleated sheets and alpha helixes, that's called tertiary structure. And then when multiple proteins come together, like in the case of hemoglobin, when we have two alpha and 2 beta subunits, then it's going to come together and make a quaternary structure. We have more than one polypeptide chain coming together to form the final protein. All right, let's talk about enzymes for a second. Enzymes are going to be biological catalysts. They bring in substrates. In this case, the substrate is sucrose. We're also using water. Remember, anytime that we're going to be breaking things down, we're going to have hydrolysis. So we're going to break sucrose down using an enzyme called sucrase. ACE is the enzyme, OS is the sugar. The products being glucose and fructose. How does that happen? Well, the active site is like a lock and key. Only sucrose can fit. Sucrose binds. The enzyme changes shape a little bit to force the separation into glucose and fructose, and then the enzyme reverts back to its native shape. So at the end, the enzyme is unchanged and free to catalyze the same reaction again on a new substrate over and over and over. Lather, rinse, repeat. So that's going to be biological catalysts, basically, and these enzymes can be regulated at multiple different levels because they're going to continue to work over and over and over until they're stopped. So they can be regulated by inhibitors that are going to bind the active site, regulated by inhibitors that are going to bind at alternate sites, also regulated at the genetic level, right? How many of these are going to be created, etc. Okay, remember, enzymes are super specific, so it's a lock and key, and they are very efficient. They're going to take something and make it happen at 1,000-fold the rate that it was happening before. So it's going to increase the rate of the chemical reaction, sometimes 10,000 or 100,000-fold. Last but not least, we'll talk about nucleic acids. We have 2 major types, DNA or deoxyribonucleic acid, and RNA or ribonucleic acid. This here is DNA. Remember, we have a major groove here and a minor groove there. So major groove is when we have this large gap, minor groove there. We're going to have a sugar phosphate backbone in purple and blue going down the outside. And then along here, we're going to have our bases. So if this were a twisted ladder, the rungs that you were standing on would be the bases, and the hand where you would be putting your handholds are going to be the sugar phosphate backbone. We're going to have our bases pairing both by size and by charge. So we have one large base pair and one small base pair binding. So adenine and guanine are both large, thymine and cysteine are both small. A is going to bind to T specifically, and G is going to bind to C specifically. How do we remember that? Because A and T are both straight lines. G and C are both curved lines. So again, we have one large and one small, and then we also are going to have hydrogen binding. Here we have 2 hydrogen binding, and there we have 3. So G and C are going to bind by sets of 3. A and T are going to bind in 2. And that's how we're going to make sure that we know that we have the exact right base pairs. All right, ribonucleic acid is going to be similar to DNA, except it's going to be a single-stranded copy of the DNA. Remember, we used the example of the long scroll with all of the recipes on it, and then I only need the cherry pie recipe, so I'm only going to make a Xerox of that particular region. That's RNA. It's going to take instructions from the genes in the cell to guide the assembly of proteins, which happens with ribosomes in the cytoplasm. All right, here, let's look at ATP. ATP is going to be our high energy carrier molecule, and if we were to remove this third phosphate, it would be ADP, or our low energy carrier molecule. And if we remove that phosphate, both those phosphates, it would be AMP, which is a cyclic messenger. It's a secondary messenger molecule. And this is the same adenine that you find in DNA and RNA. Yes, this is the same ribose that you find in RNA, but instead of it being a nucleotide, now it's going to be found as adenosine triphosphate or diphosphate, which is, again, going to be an energy carrier molecule for the cell. So those are going to be different types of nucleotides. Now let me talk quickly about the difference between DNA and RNA, right? So DNA and RNA have 3 major differences. One is going to be the sugar that's used. Deoxyribose is going to be used in DNA. Ribose is going to be used in RNA. Secondarily, going to be the base pairs that we use. So this thymine is only found in DNA. In RNA, it's going to be uracil. So A is going to bind to U in RNA. And then last but not least, it's going to ask, are you single-stranded or double-stranded? Because DNA, again, is going to be double-stranded, and RNA is going to be single-stranded. Okay, so sugar, which base are you using, and are you single-stranded or double-stranded? And that's going to become important when we decide, or the gate guards decide, whether or not this molecule is allowed out of the nuclear pores. All right, so that is the end of chapter 2. I'm going to go right on here to unit 3, talking about cellular organization. So I'm going to take just a moment here and kind of linger on this, because this is going to tell me everything I'm going to tell you about this whole chapter. So let's start here at the outside. Here, this is the plasma membrane. Plasma membrane is also going to have some things attached to it. So here we have secretory vesicles that are binding to it, that are secreting things outside, or some cytoskeletal things like microfilaments, etc., that are going to help with scaffolding and hold it all together. Inside the plasma membrane, we have the whole area that's called the cytoplasm. The cytoplasm is split up into the fluid portion, which is the cytosol, and the organelles, or the little organs. Everything that is not the nucleus is considered an organelle. Technically, the nucleus is not an organelle. It's actually the boss of the cell, right? The whole series of organelles are held together by the endomembrane system. That means that the nuclear envelope connects directly to the rough endoplasmic reticulum, which connects directly to the smooth endoplasmic reticulum, which both of those connect indirectly to the Golgi apparatus, which connects indirectly to the secretory pathways where things are going to be moving around the cell. Okay, in the nucleus we have chromosomes. Chromosomes are comprised of genes, so specific segments of the DNA molecule that code for a particular RNA molecule. The analogy that we're going to use here again are going to be the scrolls with the recipes for all of the desserts, right? The nucleus is going to have the scrolls and also the Xeroxes of the particular cherry pie recipe, which then is going to leave through the nuclear pores. The RNA leaves through the nuclear pores and find ribosomes, which can either be free or stuck here on the rough ER, and that's where we're going to enter into the site of protein synthesis. Okay, well, let's talk about the plasma membrane for a little bit. So the plasma membrane is going to be comprised of that lipid bilayer that we just talked about. It's got a lot of cholesterol molecules embedded in here. Remember, we talked about cholesterol having 3 hexamers and a pentamer. We also have membrane proteins that go all the way through. These are called integral proteins or transmembrane proteins. And then we have proteins that are going to be found only on one side or only on the other. Okay, now again, the lipid bilayer serves as a barrier function, so in order for things to come through, they're going to have to go either through these channels or through the pores or something like that. Although small molecules such as water and uncharged molecules and ions are able to come right on through. All right, let's see, different functions of the membrane protein. So again, as I talked about, proteins do a ton of different things within the cell. And I said that if everyone thought of a blue-collar job, everyone in the building could be thinking of a totally different blue-collar job and still be right, because there's so many different blue-collar jobs that are needed for Honolulu to run, or for cities to run. And that's kind of how proteins are. But we can group those by classifications, right? The same way we could group civil service jobs into whether you're helping the public in public service or whether you're helping with transportation or whatever, right? So here we have them separated into the types of proteins that they are. Are they ion channels or carrier channels. These are both integral proteins. They go all the way through, and they're going to allow things to come from one side to the other. Both of these are going to go from high concentration to low concentration. So here we're going to have ions going through this pore. We also have carriers where we have, picture this has a seat and only glucose can sit. So only the particular molecule to be transported can bind to that carrier protein, and then a conformational change happens, pulling that inside. We also have ligands and receptors. So receptors are going to be transmembrane proteins that go all the way through, and the ligand is going to be circulated on the outside of the cell. Something like a hormone might be a ligand. And when it binds, it can send intracellular information. So the ligand itself doesn't come in. Remember in class we use the example that you knock and you give a message to the butler. The butler takes the information in, but the messenger never makes it in the cell. But when the ligand binds to the receptor, that causes a conformational change inside, that causes a cascade, that sends information to the inside of the cell. All right, we can also have enzymatic proteins. We just talked about enzymes and what they do. Sometimes they're found on the outside of the cell, sometimes on the inside of the cell. They can be integral going all the way through, or they can be peripheral on one side or the other. We have linker proteins, which are functional proteins that are integral and or peripheral. And basically these are going to be structural proteins that anchor things from one side to the other. But just like the town hall has steps that, yes, you can use them for getting in and out of the building, but you can also use them for hanging out and talking about things after a case, right? So these linker and adhesion proteins are actually going to be areas where other proteins might aggregate to be able to perform cellular functions. So these perform a scaffolding role, as well as an integral role in linking the inside of the cell to the outside, etc., or one cell to the next cell, neighboring it, or the cell to the plasma membrane, etc. And last but not least, we have identity markers, glycoproteins that say, hi, I'm a heart cell, I'm a human heart cell, I'm Dr. Griffiths' human heart cell, right? These glycoproteins are going to distinguish your cells from anyone else's. And these are going to be really important for the MHC proteins, major histocompatibility proteins, right? That's an entire class of identity markers. And these are proteins that we have to kind of trick when we give individuals organ transplants. They have to take immunorejection drugs for the rest of their life, immunosuppressing drugs, because that way they're going to be able to have a organ that is non-self be disguised as self. Because when we transfer an organ in, no matter how good of a match it is, it's still going to say, hi, I'm a foreign organ. And that's what these glycoproteins do. And so that's going to be involved in the definition of self versus non-self in terms of active immunity. Okay, membranes are fluid. So they're going to have a fluid mosaic model. They're able to aggregate so receptors are able to move along their own layer. So they can't move from the inside to the outside, but along the outside they can aggregate, etc. Remember, the plasma membranes are selectively permeable. Some things can come in and other things cannot. And what comes in is dependent on what allows those things to come in. So we have transmembrane proteins like channels and transporters that allow particular molecules to come across that would not normally be able to come across. And then we have specialty molecules that are large molecules, macromolecules that are just too big that'll have to be picked up by way of vesicular transport. So we have endocytosis, or vesicular transport into the cell, exocytosis, or vesicular transport out of the cell, and transcytosis, when we can bring something into the cell and then back out of the cell, like it's just kind of passing through. Okay, so let's talk about how we get things into the cell across the plasma membrane. We can do that by either passive processes or active processes. And the difference between passive and active processes are whether or not you have to expend energy. But the passive process, you're going to go from high concentration to low concentration, and that does not require energy. But if we're going from low concentration to high concentration, it does require energy. So diffusion is the process by which particles are going to bounce off of each other in solution until eventually we end up reaching equilibrium, right? Simple diffusion happens with things like gas exchange, water exchange, etc. So we have the ability for certain things to go right on through the membrane. Remember, we talked about it being kind of like a chain link fence, and how certain smaller things, or smaller animals, can go right on through, but larger animals have to go through gates. So the smaller things that are nonpolar or hydrophobic molecules, respiratory gases, also water, even though that sounds counterintuitive, they can all go through by simple diffusion. And then this is really important for the process of gas exchange, nutrient absorption, waste excretion, etc. But we also can have facilitated diffusion, whereby things are still going from high concentration to low concentration, but they might be too large or too polar or too highly charged to move directly through the lipid bilayer. So they have to go through channels. So we have facilitated diffusion via channels and via carrier proteins. So here's a channel whereby this is potassium. Again, we're going from high concentration to low concentration, so it's secreting out. If the gate is open, we can leave. If the gate is closed, we will bounce off. So in this way, the cell can regulate its internal storage independent of what's going on the outside. And if it wants to release things, it's able to open the gate and allow potassium out. But if it has too little potassium and it wants to keep the potassium in, it's able to close the gate and keep the potassium inside the cell. Now, here is a transporter molecule. Here's a glucose transporter. Again, I want to be very clear. This is specific to what's allowed to sit here. Fructose cannot sit here, right? Not only can not just any molecule, not just any sugar, only this sugar can sit here. And once glucose binds, we have a conformational change whereby glucose is imported into the cell. Again, it's going from high concentration to low concentration. This does not cost energy. But like the example I gave in class where we are a bunch of people that are waiting to cross the street, when the light changes, although we're all going with the gradient, some people might need assistance. Small children might need you to hold their hand. Elderly people or people with a wheelchair might need someone to push them across the street, right? And so that's what's happening here. We're still going from high concentration to low concentration, but this is offering someone a place to sit down to come across the membrane, all right? So this is an overview of what we're seeing. So we're always going from high to low concentration if we're going to be doing passive transport. Examples include simple diffusion, water, small gases, things that are able to make it through that chain link fence. Or we can go through via a channel, or we can go through via carrier. Again, most individuals are going to be able to walk across the street on their own, but some of them are going to need assistance, and they can do that either by channels or by carriers. The next thing of diffusion is for osmosis. Osmosis is the selective movement of water. Remember that semi-permeable membrane is going to allow water across, but some things might not be allowed across, like solutes. So in this case, we can look at the concentration of solute molecules and immediately flip it around to the concentration of water molecules. So if we have 0% solute and 5% solute, what I want you to immediately think is 100% water and 95% water. Water is going to go from high concentration to low concentration. And because that solute can't go back across, we're never going to reach equilibrium, because no matter how much water we put into that 95% solution, it's never going to reach 100%, right? So that means we're going to have pressure. Via hydrostatic pressure, we're going to have movement over. And if we want to figure out what that highest hydrostatic pressure, also known as osmotic pressure, equates to, we can put an applied pressure down until the volumes are equal. And when we do that, whatever that applied pressure is, it's going to equal the osmotic pressure, or the force of water from one side of the membrane to the other to try to reach equilibrium. Okay, and this is going to bring me to my next slide. Here we have a blood cell that's either in isotonic, hypotonic, or hypertonic solution. So in isotonic solution, we have the same concentration inside and outside. So say we have 5% inside the cell to make it easy. We also have 5% outside the cell. So again, flip it into water, 95% water, 95% water, even, isotonic. Now, that doesn't mean we're not having water motion. We're still having water move in and water move out, but the point is that the net motion of water is equal. Now in a hypotonic solution, now we have a 5% salt inside the cell and 0% salt outside the cell. Again, what do I want to turn that into? 100% water and 95% water. So which direction does the water go? Into the cell, starts swelling up like a hippo. So a cell put in hypotonic solution swells up like a hippo. And if we put it in hypertonic solution, say we put it in 10% salt. 10% salt outside, 5% salt inside. Again, I want you to immediately say that becomes 90 and 95. From a high concentration to low concentration, mean water's leaving the 95%, going into the 90%. So cells that are put in a hypertonic solution are going to purge all of their fluids. Okay, so the tonicity of solution is really important because that's going to influence how the body cells are going to interact. Are they going to lyse? Are they going to shrivel? Or are they going to stay in normal shape? All right. All of that was referring to passive processes. Now let's talk about active processes or active transport. So for active transport, we have primary active transport and secondary active transport. Active transport requires energy to move solutes against a concentration gradient. So we're going from low to high here, and it's going to cost you cellular energy. For primary active transport, that cellular energy is going to be in the form of ATP. So here we have the sodium potassium transport pump. So those 3 sodium ions are going to get pumped out and 2 potassium are going to get pumped back in. You can see there's a conformational change depending on which way it's facing. In order for those 3 to get pumped out, ATP is going to have the free phosphate popped off of it, and ADP, or low energy version, is going to be released. That phosphate's going to stay put until those 2 potassium molecules come back in, and then that free phosphate is released. The overall difference is that we have 2 potassium in, 3 sodium out. So now we've got a chemical gradient and electrical gradient, right? So we've got a gradient for sodium and a gradient for potassium. And now we have expelled 3 positive and only brought in 2. So now we have a positive charge gradient as well. All right, so that's primary active transport. Now let's look at secondary active transport. So secondary active transport still requires energy, but we're being sneaky about it. So we're not going to use ATP. We're actually going to be using the gradient of another molecule and hitching a ride. So in this case, here's our sodium. Remember, we had that sodium potassium pump, so we had all that sodium pumped out. Now we're going to use the sodium coming in Sodium gradient is going this direction. And when the sodium comes in this case, we're pulling something else with it, such as glucose or an amino acid. Those are examples of symporters. We're riding or hitching a ride on this, which is already not requiring energy. And then we're bringing in something that does require energy kind of on that flow of this gradient. This is the equivalent of saying, I'll come into the party only if you let my friends in too. And the opposite can happen with an antiporter. I'll come into the party only if you kick my ex-husband out, right? So in this case, sodium is coming in along the gradient and calcium is being pumped out against the gradient. Again, it does not cost us any energy because we are using the energy flow from going with the gradient to drive something that's going against the gradient. All right, so now let's talk about transport. Okay, so we can have transport of things into the cell. That's called endocytosis. We have a couple different types of endocytosis. So this is going to be receptor-mediated endocytosis. So here with receptor-mediated endocytosis, we have these little LDL that we're bringing in. We have LDL receptors. LDL binds the receptor, gets its little seat on the bus. At the same time that clathrin is a molecule on the inside of the membrane. And you can picture this kind of like small triangles. So many small triangles together would eventually form an invagination and eventually form this circle, right? So this is going to be called invagination of the vesicle. The vesicle is then going to get decoded. So a clathrin-coated vesicle is then going to get uncoated. The clathrin is going to go back up to make a new clathrin-coated pit. And also we're going to deseat or remove the molecules off of their receptors. So the LDL molecules comes off of those LDL receptors. Those LDL receptors get recycled back up to the surface. And again, lather, rinse, repeat. So in this way, we can bring something in that we're looking for from the external environment by putting receptors out, grabbing the individual molecule and pulling it into the cell. Okay? And again, once we bring it in, there's a couple things we can do with it. Mainly what we're going to do with it is degrade it down into its biological Lego pieces, right? The smallest parts that we can use, the building blocks that we can use to build our own biomolecules. But we also, whatever's left over is going to form a residual body, which is going to end up expelled because we don't want the waste to end up inside the cell. Sometimes that residual body can first hit up a peroxisome where it can be degraded so that we don't have anything like biological toxins that are going to end up being released into the extracellular fluid. All right, another type of endocytosis is called phagocytosis, or the ingestion of solid particles. Again, it's going to be receptor-based. These receptors are going to reach out and grab these microbes and pull them in. So pseudopods are going to reach around the microbe. The receptors are going to be on the inside of the pseudopods, binding the microbe and pulling it in. This takes less than a minute. Here's a white blood cell engulfing a microbe. And then after we engulf it, we're going to fuse it with a lysosome. And then eventually we're going to have a digestion. Then the residual body can do one of two things. It can go to a peroxisome to be completely decontaminated, or it can just be released out at the surface of the cell. All right, another thing is called pinocytosis or cell sipping, which I guess is not written here, but basically it's an ingestion of intracellular fluid. Here, yeah. The ingestion of intracellular fluid, which is then going to fuse again with the lysosomes, and in this way we're going to digest it and then be able to release those solutes into the cell for us to be able to use. Exocytosis is kind of the opposite of all of that endocytosis. It's basically going to be when we have a structure called a secretory vesicle that's inside the cell. It's going to fuse with the plasma membrane and release the contents to the extracellular fluid. We can also have, again, transcytosis, which is something when a vessel can come into from one side and then exit out the other side without depositing any material inside the cell itself, just kind of using the cell as a byway or through that cell layer to get to the next cell layer. Okay, again, the cytoplasm is split up into the cytosol and the organelles. Cytosol is the intracellular fluid, and the organelles are going to include multiple things, including the cytoskeleton. So the cytoskeleton is going to be a protein fiber, protein filament fiber. We have microfilaments, intermediate filaments, and microtubules. Microfilaments are going to be found at the outside of the cell. These are kind of structural proteins that help scaffold to push the cell outward. Intermediate filaments are found within the cell. And the microtubules are found in multiple different regions, but these are going to be involved in locomotion. So these are going to be involved in mitosis. So we'll talk about that when we get to mitosis. And they also are going to be involved in migration. So moving a cell from one piece to another, for example, like through flagellar or ciliar motion. Okay. Other types of organelles, here's a centrosome. So a centrosome is going to have a triplet, a microtubule triplet. And if you picture that this one contracts and then that one contracts and then this one contracts, right, alternating, we might be able to have motion. That would be what we're going to see when we look in cilia and flagella. But that same structure is also found inside a centrosome. Centrosomes are going to be found during the process of mitosis. We're going to have one migrate to the one side of the cell, one migrate to the other side of the cell. They're going to form spindle fibers, which connect at the centromeres of the chromosomes during the metaphase plate. At that point, when we head into anaphase, the centrosomes are going to start pulling the spindle fibers closer to each other and separating out the sister chromatids. All right, so cilia and flagella are specialty organelles that are meant to either, for cilia, move fluid across the surface of the cell, or for flagella, propel the cell through a liquid medium. You can see here ciliary motion with a power stroke and a recovery stroke. Flagellar motion is just going to allow us to move forward. Again, you can remember that that works because of these microtubule triplets, which are pulling left, right in concert. All right, ribosomes are going to be the site of protein assembly. We have a large subunit and a small subunit. The small subunit is going to wait for the mRNA to bind. Once the mRNA binds, the ribbon of mRNA binds first, then the large subunit binds on top to make a functional ribosome. It's going to move along one codon at a time, which is one set of 3 nucleotides, which is going to then compose 1 amino acid. Endoplasmic reticulum is part of the intermembrane system, the endomembrane system, and basically it's going to be a series of membranes that form these flattened sacs or tubules. They're called cisternae, and inside we can either have ribosomes for the rough ER or no ribosomes for the smooth ER. The ribosomes on the rough ER are going to be a site of protein synthesis, protein assembly, and then here in the smooth ER, this is where we're going to be making our lipids. The Golgi complex is going to be our sorting mechanism. Remember the example I used was dropping a letter in Kansas. At first it has to go either west or east, then it has to go either north or south, then from there it has to go to the state, then it has to go to the city, then it has to go to the neighborhood, then the street, then the address. So each of these layers is going to increase in complexity in terms of sorting. So a protein is going to have multiple little tags on them, and as it travels from layer 1 to layer 2, the next tag is going to be revealed, determining how close or where it's going to go for each of the layers. Eventually you're going to end up with secretory vesicles that are all going to one specific address. So again, this is when we're going to separate into north and south, and then east and then west, and then this state, then this city and that city. Eventually we get to this suburb, eventually get to this address. And these secretory vesicles are going to contain proteins that are all headed to the same location, whether that be the lysosome or the plasma membrane or whatnot. Okay, so again, that looks like this. We have specialty tags on those synthesized proteins. Those tags are going to then help with the sorting at each layer until eventually we end up with vesicles that are specialty vesicles that are headed to specialty locations like the lysosome, the membrane, secretion, transfer, yada, yada. Okay, we also have some recycling and trash cans. So that's going to be the lysosomes, the peroxisomes, and the proteasomes. Lysosomes are going to be a catch-all recycling center that have very powerful digestive enzymes. They take both recycling and trash, and they're going to digest everything that they can. And when they can't, they make residual bodies. Residual bodies are going to bind to peroxisomes. Peroxisomes are a little bit smaller but have chemical structures in there that are not just digestion but are oxidases that are meant to oxidize substances. Eventually we're going to be destroying those substances, and then we can release them to be picked up, whatever's left over to be picked up as extracellular waste. Proteasomes are recycling centers that are only for cans, or in this case only for proteins. So it's a really specialty type of a recycling center that destroy unneeded, damaged, or faulty proteins that are created by the cell to be able to create small polypeptides that they're able to then use later on to be able to create new proteins, right? Mitochondria is the powerhouse of the cell. This is where the cellular respiration electron transport chain is going to take place. Again, that external membrane is going to be specific to the host. So if it's in a dog, it's going to be a dog membrane. Human, it's going to be a human membrane. But that inner membrane is a mitochondrial membrane with specific mitochondrial characteristics. We also have cristae, which are going to go in between, so the folds in between there. And this is going to be the site of ATP assembly, so cellular respiration. Basically what we're doing is we're taking a glucose molecule, we're breaking it down. That's an ordered 6-carbon molecule, and we're breaking it down into 6 simple carbon dioxide molecules and water. And we're doing that, when we do that, we release energy. We're capturing that energy in the form of ATP. So mitochondria are really important in terms of ATP production. The nucleus, again, this is not an organelle, although when I was in school it was considered an organelle, so I apologize if I say that it is. It is not considered an organelle. It's a very prominent cell feature, which is going to be responsible for the creation of everything in the cell. We have the nucleolus in the center, which is the site of ribosome assembly. Then we have the chromatin, which is how our cells normally are going to have our DNA. Remember, this is the equivalent of a woman having her hair down versus pulled back up, right? You can't brush it when it's pulled back up, which is how our chromatin is going to condense down into chromosomes during division. But most of the time it's accessible. Right, back to that scroll example, you want to be able to make the Xerox of the scroll. You don't want the scrolls to be wound so tightly that you can't access them. So again, the chromatin is going to make RNA from DNA. Once the RNA is created, it's going to bounce off randomly against the nuclear pores and be asked 3 questions. What's your sugar? What's your base? So do you use uracilthymine? And are you single-stranded or double-stranded? If you answer all of these questions correctly, you're going to exit out through the nuclear pore, and you're going to end up being able to find a ribosome, which is going to be the site of protein synthesis. Okay, so now let's talk about condensation. So here we have chromosomes. Again, this is the DNA that we saw before with the nucleotides, the base pairs across the center, sugar phosphate backbone shown in purple. They wrap around histones. Histones are an octet. Wraps one set of DNA wraps 2.5 times around each histones, and those histones coil on each other. Then they supercoil. Then they make loops, then eventually forming that characteristic structure that you recognize as a chromosome. Here at the center, we have the centromere. The middle of the centromere is going to have a region called a kinetochore. This is where our spindle fibers are going to bind during the process of mitosis and meiosis. All right. So now let's talk about protein synthesis. Two major steps. First, the process of transcription, which is when we take DNA and we make RNA out of it. Remember I talked about monks copying the Bible from Latin into Latin. So keeping the same language of nucleic acids into nucleic acids is considered transcription. But when we go from Latin into English, that's translation. So now we're going from nucleic acids into amino acids. And again, that happens in the cytoplasm with the help of ribosomes. Okay, so let's look at the first step. The first step is going to be the creation of RNA from DNA. So to do that, we use something called RNA polymerase. RNA polymerase is going to unwind the DNA in front of it and also bring in the matching nucleotide. So if we have an A, it'll bring in a U. If we have a T, it'll bring in an A G, it'll bring in a C, right? So that we can then have this elongating chain. Eventually, it'll get processed. So this region is an intron. It'll be snipped out, and then these two will be spliced together. Picture that you taped an entire TV program, but you want to edit out the commercials, so you'll snip those out. Then we'll end up with a long strand of RNA, which can leave the nucleus through the nuclear pore. Why? Because we're asking the right questions. Are you single-stranded or double-stranded? What is your sugar? And are you using uracil or thymine? If the answers are uracil, single-stranded, and ribose, then you're allowed out of that nuclear pore to find the site of protein synthesis in the cytoplasm, which would obviously be on the ribosomes. The ribosomes are found on the rough ER and also free in the cytoplasm. So in order for translation to occur, first we need that mRNA molecule, which we just had leave the nuclear pore. It has to bind to the small subunit. Once it does bind, then we're going to have the large subunit binding. Inside the large subunit, we have three regions: the access site, the peptide bond site, or the P site, and the exit site. Access is where we're going to be bringing in our new tRNA molecule, and exit is where our old tRNA molecule is going to be leaving. All right, so let's start here with one. First, we have the small subunit, and the mRNA. Then, where it's going to stay open until a tRNA molecule. Remember, tRNA molecules look like little guns. They have anticodon regions, and they also have amino acids. So one section is going to have an anticodon that binds to the mRNA, and one section is going to have an amino acid, which will get added to the growing, elongating polypeptide chain, right? Okay, the very first codon-anticodon interaction is always going to code for methionine because the start codon always has the methionine amino acid. When that happens, the large subunit is going to bind. Now, if the large subunit binds with the methionine amino acid, tRNA containing amino acid thionine in that middle space, now we have this codon exposed in the access site. A bunch of different tRNA are going to try to bind. Only the ones that have the right anticodon region will bind. When that right one does bind, then that amino acid is then going to get added from the developing chain, and the whole thing shifts over. When it shifts over, the one that's in the exit site gets released, and a new one is going to come into the access site, and in this way, the developing polypeptide chain is going to get longer and longer and longer. It's also of note that we're going to have multiple different ribosomes working on the same mRNA transcript. So this one is waiting to bind, so we only have the small subunit. This one is just starting. This one's getting longer and longer and longer, and eventually this one has dissociated and made a complete protein. So in this way, we can be making many protein copies from the same RNA transcript at the same time because the mRNA molecules are just following in succession of one another. Okay, so again, what do we need for this to work? We need the mRNA. We need the small and the large ribosomal subunits. We need the tRNA with the anticoding regions that are going to bind to each of them. And then, of course, the tRNA is also going to need the amino acids to be able to create that polypeptide chain. Okay, so now let's enter into cell division. Remember, cell division is split up into interphase and mitosis. Now, not all cells are going to be in one or the other. We can also enter into G0, which would be exit out of the cell division for non-dividing cells. Interphase is split up into three main sections: the G phase, the S phase, and the G2 phase. G phases are the growth phases. S phase is the synthesis phase. There are going to be stop points and checkpoints as we go along here. because this is going to be the point of no return. But let me I digress for a second. So G1 is when we are duplicating our organelles and cytosolic components, and our centrosomes are starting to replicate. Once we pass that checkpoint and we say, yes, your DNA has been acceptable for replication, then we're going to enter into the S phase where the DNA is going to be replicated. This takes about 8 hours. This is the point of no return. If your DNA is replicated but you can't go into G2, you're going to enter into apoptosis, or programmed cell death. Again, this is different from necrosis because it's a structured deconstruction of the cell. So if, for example, I knew that I was leaving forever and I told all my neighbors, you know what, if you want my marble countertops, come take them. You want my stained glass windows, come take them. And so all my neighbors came and deconstructed my house. That's much different than just letting the elements come and take over it, which is something like necrosis. All right, I digress. So we've passed this checkpoint. The DNA has been replicated and we can enter into G2. More cell growth is going to happen. We're going to have the synthesis of many enzymes and proteins. And then we're going to enter into mitosis. Mitosis is split up into prophase, metaphase, anaphase, and telophase. And I like to liken this to a play. This is setting the stage. This is opening the curtain. This is the end of the show, and this is the close of the curtain, right? So cell division has multiple stages. Prophase is when we are setting up the stage. Oh, I apologize. Let's talk about interphase. Again, interphase, we have G1, S phase, and G2. G1 is when we are making duplications of our organelles in our cytoplasm. S phase is when we're copying our DNA. And G2, we're going to have the continuation of cell growth as we're preparing for the mitotic event. Okay, so mitosis is going to involve major steps include prophase. First step of prophase is going to be the dissociation of that nuclear envelope. So you can see the nuclear envelope is breaking down. Our chromosomes are going to start condensing. So previously it was chromatin. And the chromatin, remember this lady's hair down, now it's getting braided and braided and braided, and so that's nice and tight in pretty little chromosomal fashions. And then they're going to be getting ready for the metaphase spread. So prophase has an early and a late. Anaphase also has early and a late. And in the early stages, we still have a nuclear envelope and still have some chromatin. That's here. In the late stages, we're condensing pretty much entirely into chromosomes, and we have no nuclear envelope. We are now going to be heading into metaphase. During metaphase, all of the centromeres line up in the exact same place in the equatorial plate. Spindle fibers come across and bind to the centromeres at the kinetochore. And now we're getting ready to start pulling them apart. As soon as we start pulling them apart, that's anaphase. So early anaphase is the beginning of the pulling apart of the sister chromatids. Late anaphase is when we are pulling them apart to the point where we're at the opposite poles, and we are just about to enter into telophase. So telophase begins as soon as the chromatid movement stops, and we're going to start reforming the nuclear envelope, and the chromosomes are going to start decondensing into chromatin. Okay? Right after telophase, we go into cytokinesis. And telophase and cytokinesis are akin to walking out a door and the door shutting. Yeah, they happen in order that you walk out first and then the door shuts, but they happen almost at the same time. Cytokinesis happens 1 second after telophase. Basically, we're going to have a cleavage furrow that's starting during telophase, and eventually it's going to pull itself down into 2 separate cells. So cytokinesis is just splitting into 2 cells. And then that's going to end up with 2 identical cells, which are now back into interphase. Okay, so at the end of cytokinesis, right, we're going to have telophase splitting us into 2 cells in interphase. All right, so cells have 3 major possible destinations or fates, right? Not destinations, destinies, I apologize. 3 major destinies. They can either continue to function without dividing, remain alive, so that's G0, basically stay where you are, differentiated as is, and never do anything else for a while. Or we can grow and divide, or you can die off. Again, we have 2 different types of cell death. We have programmed cell death called apoptosis, which is like a cell suicide gene. This is a very structured cellular deconstruction, whereby things that are taken from that cell are utilized by the surrounding cells. And then we have necrosis, which is a pathological cell death. Basically, the cell's starving to death. It's not getting its nutrients and it's drowning in its own waste. It's due to injury, and it's basically akin to me abandoning the house and letting nature take over instead of allowing my neighbors to take it apart. All right, so that's meiosis. That's mitosis. Now let's talk about meiosis. Meiosis has 2 major stages. So unlike mitosis, where we made 1 replication event and 1 division event, with meiosis, we have 1 replication event and 2 division events. So before I jump in here, I want to talk about the major differences between meiosis and mitosis. So with mitosis, you have 1 division event that results in 2 genetically identical diploid cells. The cells are identical to their parental cell line, and they have 2 copies of every chromosome. In meiosis, we end up with 4 genetically unique haploid cells, cells that have half of the genetic material of their parental cells and are completely genetically unique both from their parents and from their other sibling or offspring cells. All right, however, we still have the same major stages that occurred during mitosis. So we're going to have prophase, metaphase, anaphase, and telophase, except then we're going to have prophase 2, metaphase 2, anaphase 2, and telophase 2. So something special happens during prophase 1, and this is called recombination. So basically what happens if I'm the one that's undergoing meiosis, that means that I'm in the process of creating a mature egg. And in order to do that, first we have to have crossing over. So my maternal alleles and my paternal alleles, that means my mom's chromosomes and my dad's chromosomes are going to align, and we're going to have a crossing over event. Now remember that my mom cooks with cinnamon and my dad cooks with nutmeg. So although we have the same pies, right, blueberry pie, apple pie, it's in the same order, the alleles, that is, are in the same order on the chromosome, we're going to be switching the versions of the recipes. So now we're switching, this one is nutmeg for cinnamon. And now, so this one is all cinnamon, this one is all nutmeg, this one is half cinnamon, half nutmeg, this one is half nutmeg, half cinnamon. In this way, we have shuffled the grandparents' alleles prior to creating, right? So if I'm the one creating the egg, it's my parental alleles that are my mom and my dad's alleles that are being shuffled prior to be making the egg, which is then going to be fertilized to create my offspring. So really can consider this like grandparental alleles that are shuffling. But my point is that we actually are going to inherit chromosomes that are pieces or fragments of all of our ancestors' chromosomes, because our chromosomes have been shuffled so many times during this process. So that's kind of cool to think of. All right, so during prophase 1, after we have the recombination event, the sister chromatids are going to line up during a metaphase plate. So during metaphase 1, then anaphase 1, they're going to be pulled to opposite sides. Telophase 1, we're going to have them separated to opposite sections. However, during telophase 1, oh, I'm way back. Sorry, I shouldn't have hit down. I should have used this. Here we go. So during telophase 1, instead of our nuclear envelope reforming and our chromosomes decondensing, we pretty much are going to slip straight from telophase 1 into prophase 2. So we're not going to necessarily have our nuclear envelope reform. There's no point. We're just going to enter into a second set of meiotic events. And from there, telophase 1 into prophase 1, so notice there is a cytokinesis event. And then these cells will then eventually enter into a second set of metaphase 2, anaphase 2, and telophase 2. Again, the end result of this is that we have haploid cells, so they have one chromatid of each, right? They have one, instead of being diploid, they are haploid. So we have 4 haploid cells that are genetically unique as opposed to 2 diploid cells that are genetically identical. Okay, so one of the last things I want to leave you with is the idea that although all of these cells might come from the same genetic material, right, from the same individual biologically, obviously we're talking about a male here, right? This cell is haploid and only has half of the DNA. This cell is anucleatic. It doesn't have any DNA whatsoever. These 2 cells have the same DNA. These 3 cells have the same DNA, but they're expressed differently, right? The nervous cell is going to have nervous DNA expressed, nervous tissue DNA. This is going to have muscular DNA expressed, epithelial DNA expressed. And so because of the expression of the genetics, we're going to get different outward appearance of the cells even though they have the exact same nuclear makeup, okay? So the shapes of the cells, the size of the shells, the usage of the cells is going to vary considerably, even given the same genetic construction, okay? And we're not going to focus too much on aging and free radicals and disorders, etc. So I am going to wrap it up there. Thank you so very much for listening to my brief lecture on Exam 1's review, and I hope that you guys have a fantastic exam. Reach out to me with any questions you might have. My cell phone number is listed in the syllabus, so go ahead and text me with any questions or concerns that you might have. Have a great day, guys, and happy studying. Aloha.